Okay, because we're going to look at translation, which is the converting of the genetic code into protein. But before we do, I want to talk to you a little bit about codons and the genetic code chart. So I'm going to use the same messenger RNA that we made during transcription. So I'm just going to use the same one, and I'm using a brand new sheet of paper to do this. So I'm just copying this over from our notes. So this is our messenger RNA that we transcribed from a piece of DNA in the nucleus, and then it detached from the DNA, and then the DNA, the double helix, can go back together, and it has traveled out of the nucleus to the ribosomes to be translated into the proteins that we need. Okay, so this is basically our genetic code that we're working with. So this is a genetic sentence. So when we're talking about a genetic sentence, we're talking about genetic code words. And each genetic code word is only made up of 3 nitrogen bases, or we could say 3 nucleotides, and they're consecutive, so one right after the other. So each of these is going to code for an amino acid. So the name that's given to these is a codon. So this little genetic sentence here, this little piece of messenger RNA has 2 codons. So that means that this gene only has 2 amino acids. So a little bit here, I'm going to write down just a little bit of information about the codon itself. Codons, genetic code words. And they code for amino acids. Now, when it comes to the amino acids, there are 20 amino acids. And if I take these 4 different letters of the RNA alphabet, remember, is AUCG. If I take those 4 little letters and I rearrange them in sets of 3s, then what I'll end up getting is 64 different codons. So what that means is that some amino acids will have more than one genetic code word. Alright, so now I'm going to show you how to use this genetic code chart. Now there's a couple of different versions of this chart, but both of them work the same way. It's all about taking the genetic code, and basically we're deciphering this code and determining what amino acids that it codes for. And these amino acids will build our proteins. So I've got 2 different versions here, and I'll provide you with a copy of both versions. There's this one. This looks more like a circle than a chart. Oh, it's a wheel. And then there's this one, which definitely looks more chartish. So what I'm going to show you here is how to use this bad boy. Now we have a very small little gene here. We've got AUG and UGA. So with this guy, the one that's the wheel, and you can see here this key, it reads from the inside out. So the inside, we can see we have it divided up in 4 quadrants, and each of them has one of the letters from the RNA alphabet. So if you notice, there is 1, 2, 3 different levels of letters. So this one represents the first, second, and third letter in your codon. So if I'm trying to decode AUG, I'm going to start here with the A. Here's my A. And then I want to go to my U and then to my G. So this little pathway here, try to make that as beautiful as possible. Oh yeah. Leads us to this abbreviation, MET. And this stands for the amino acid of methionine. Now there's something else about this MET. So I'm going to show you on this other chart, the chart, which also has its own little key here. The darker parts, you can see there, is the chain termination codon. We call them stop codons. And the lighter place is the initiation codon, which we don't call it the initiation codon. We just call it the start codon. So just to show you how this one works, it works a little differently. Here is my first letter of my codon, second letter, and third letter. So again, I'm trying to find AUG and what it codes for. So here's my A. My second letter is U. So I'm here. And my third letter is G. So where they all converge is at methionine again, and we also see here that this is also referred to as the start codon. So that means it's the start of this gene that codes for a protein. So on our note sheet here, we need to make sure that we include that. So AUG, it does stand for the amino acid methionine, but it is our start codon. So it's the beginning of our gene. And an easy way to remember it, AUG, the start of school. We start school in August. Okay, we have one other little codon here that we'll play around with. So I'm going to go back to my genetic code wheel, my wheelie chart, and now I'm going to find UGA. So there's my first, second, and third base. So U, oh lovely already, G, and then A. And this is known as the stop codon. So this is like the period at the end of a sentence. Now if we look at it using our other traditional chart, so here's our first base, which is the U, and now our second base is the G, so now we're here, and our third is the A. And where they all come together is at UGA, which is the termination sequence. It's the terminator. And it just means that when you're translating, that the process of translation, the making of that protein, will stop when it reaches one of the three stop sequences. So this is the end of the gene.